These therapies have shown promising results in treating cancers such as melanoma, lung cancer, and non-Hodgkin lymphoma. 3. Autoimmunity and immune tolerance. In autoimmune diseases, secondary immune modulation is critical to limit the pathological activation of self-reactive immune cells. Strategies like immune tolerance induction, which promote TREG expansion and inhibit effector T-cell activity, are being investigated for conditions such as rheumatoid arthritis, type 1 diabetes, and multiple sclerosis. 4. Chronic infection and immune exhaustion. In chronic infections like HIV, hepatitis, or tuberculosis, the immune system experiences T-cell exhaustion due to prolonged antigen stimulation. Secondary immune modulation in these cases focuses on reviving exhausted T-cells using immune checkpoint inhibitors or cytokine therapies. Primary versus secondary immune modulation. Feature primary immune modulation, secondary immune modulation. Stage of immune response: initial antigen exposure and activation of naive T B cells, fine-tuning of effector response and memory formation. Key players: antigen-presenting cells, naive T B cells, cytokines, effector T B cells, memory cells, regulatory T cells, immune checkpoints, outcome activation of adaptive immunity, long-term immunity, regulation of immune response. CSIR-NET previous year questions. 1. CSIR-NET June 2020. Which of the following immune mechanisms is primarily responsible for T-cell exhaustion during chronic viral infections? A. Increased IL-10 production. B. Immune checkpoint activation PD-1, PD-L1. C. Lack of cytokine secretion by APCs T-Treg depletion. Answer: B. Immune checkpoint activation PD-1, PD-L1. 2. CSIR-NET Deck 2019. In secondary immune responses, the role of memory B cells is to approach high-affinity antibodies more rapidly than naive B cells. B migrate to lymph nodes to present antigens to T cells. C secrete large amounts of cytokines to activate T cells. D induce T cell apoptosis upon antigen re-exposure. Answer: approach high-affinity antibodies more rapidly than naive B cells. 3. CSIR-NET June 2018. Which of the following cytokines plays a key role in the regulation of immune responses by promoting the expansion of regulatory T cells? TREGs. A. IL-12. B. IL-6 circuit TGF beta. D. IL-2. Answer: C. TGF beta. Conclusion. Secondary immune modulation is critical for ensuring immune homeostasis and memory responses. After the primary immune activation, this modulation ensures that the immune system remains poised to respond effectively to subsequent infections while preventing overreaction or autoimmunity. The interplay between memory T and B cells, cytokine signaling, and immune checkpoints is essential in shaping the outcome of both adaptive immune responses and immunotherapies. This topic is a high-yield area for CSIR-NET life sciences, especially for understanding the mechanisms of immune regulation and their clinical applications. Let me know if you'd like the next lecture on immune memory and vaccination or cytokine signaling and immune responses. Complement system, ChatGPT said. Here is your CSIR-NET JRF Life Sciences level lecture on the complement system, written in maximum detail with paragraph format, aligned with CSIR-NET exam standards. The lecture includes 2 to 3 previous year questions with answers and years mentioned and is curated as your only revision and study material. Complement system, CSIR-NET Life Sciences lecture introduction. The complement system is a crucial part of the innate immune response that enhances the body's ability to fight infections. It consists of a series of proteins that circulate in the blood and are activated in a cascade-like fashion upon detection of pathogens. These proteins work together to eliminate pathogens through various mechanisms, including opsonization, cell lysis, and inflammation. The complement system acts as a bridge between the innate and adaptive immune systems and plays a vital role in immune surveillance, inflammation, and pathogen elimination. Components of the complement system. The complement system comprises over 30 proteins that are mainly produced by the liver and circulate in an inactive form. Upon activation, these proteins undergo proteolytic cleavage and form an enzymatic cascade that leads to the formation of active complement fragments. The major components include 1. Complement proteins C1 to C9. These proteins are designated as C1, C2, C3, C4, C5, C6, C7, C8, and C9. Upon activation, the complement proteins are cleaved into small and large fragments, e.g., C3a and C3b, that perform various immune functions. C1, the first component of the classical pathway that binds to antibody-antigen complexes or pathogens. C2, C4, involved in the formation of the C3 convertase during classical and lectin pathways. C3, the central protein in the complement cascade that can be cleaved into C3a, an anaphylotoxin, and C3b, which participates in opsonization. Plex MAC, which creates pores in the pathogen's membrane, lead to cell. 2. Regulatory proteins. These proteins prevent excessive complement activation and host tissue damage. Some key regulatory proteins include CD55 decay accelerating factor, DOF, inhibits the formation of C3 and C5 convertases. CD59 protectin prevents the formation of the membrane attack complex on host cells. Factor H and factor I inhibit the alternative pathway by binding to C3b and accelerating its decay. Activation pathways. The complement system can be activated through 3 main pathways, which converge at the activation of C3. Each pathway involves different initial recognition mechanisms but leads to a common terminal pathway that forms the membrane attack complex MAC. 1. Classical pathway. The classical pathway is activated when antibodies, or IgM, bind to antigens on the surface of pathogens. This binding activates the C1 complex composed of C1q, C1r, and C1s, which then cleave C2 and C4. The C4b-C2a complex form serves as a C3 convertase, which cleaves C3 into C3a and C3b. C3b can then bind to the pathogen surface and form a C5 convertase, which activates C5. 2. Lectin pathway. The lectin pathway is activated by the binding of mannose binding lectin, MBL, or Facolin-specific carbohydrate patterns on the surface of pathogens. This binding leads to the activation of MASP's MBL-associated serine proteases, which cleave C4 and C2, forming the C3 convertase similar to the classical pathway. This pathway does not require antibodies and is primarily involved in innate immunity. 3. Alternative pathway. The alternative pathway can be initiated spontaneously through the hydrolysis of C3, forming C3H2O. This complex can bind to factor B, which is then cleaved by factor D to form C3BBB, the alternative pathway C3 convertase. The alternative pathway is continually activated at low levels, a process called tickover, and serves as an amplification loop for the complement cascade. Functions of the complement system. The complement system performs several critical immune functions. 1. Opsonization. C3b binds to the surface of pathogens, marking them for recognition and ingestion by phagocytes such as macrophages and neutrophils. This enhances the phagocytic process by promoting the binding of the pathogen to the complement receptors, e.g., CR1 on phagocytes. 2. Inflammation. Anaphylotoxin activation. The small fragments C3a, C4a, and C5a are anaphylotoxins that promote inflammation by inducing mast cell degranulation, releasing histamine and other mediators, increasing vascular permeability, allowing immune cells and proteins to enter the site of infection, attracting immune cells like neutrophils and monocytes to the infection site via chemotaxis. 3. Membrane attack complex MAC formation. The terminal pathway of the complement system involves the assembly of C5b, C6, C7, C8, and C9 to form the membrane attack complex MAC. The MAC forms pores in the pathogen's membrane, leading to cell lysis. This is particularly effective against gram-negative bacteria and some viruses. 4. Immune complex clearance. The complement system facilitates the clearance of immune complexes, antigen-antibody complexes, from circulation. C3b can bind to immune complexes, and these complexes are then cleared by phagocytes, preventing tissue damage due to accumulation of immune complexes. Regulation of the complement system. Excessive activation of the complement system can lead to tissue damage and autoimmune diseases. Therefore, the complement system is tightly regulated by various regulatory proteins. C1 inhibitor C1INH inhibits the classical pathway by binding to C1R and C1s, preventing them from cleaving C2 and C4. Factor H and factor I inhibit the alternative pathway by binding to C3b and facilitating its degradation. CD59 protectin prevents MAC formation on host cells by binding to C5b 678, blocking the recruitment of C9. Clinical relevance of the complement system. 1. Immune deficiencies. Defects in complement proteins can lead to increased susceptibility to infections. For example, C3 deficiency leads to susceptibility to pyogenic infections. C1 inhibitor deficiency causes hereditary angioedema, a disorder characterized by recurrent episodes of swelling. 2. Autoimmune diseases. Overactivation of the complement system is implicated in several autoimmune diseases. Systemic lupus erythematosus (SLE) is associated with the deposition of immune complexes and activation of the complement system, leading to tissue damage. Rheumatoid arthritis (RA) can be exacerbated by complement activation, leading to chronic inflammation in the joints. 3. Therapeutic targeting. Complement proteins are also being targeted in therapy. Eculizumab, a monoclonal antibody targeting C5, is used to treat paroxysmal nocturnal hemoglobinuria (PNH) and AHS atypical hemolytic uremic syndrome. CSIR-NET previous year questions. 1. CSIR-NET DEC 2020. Which of the following is not a function of the complement system? A. Opsonization. B. Toxin neutralization. C. Inflammation. D. Antigen presentation. Answer. D. Antigen presentation. 2. CSIR-NET June 2019. The formation of the membrane attack complex MAC in the complement cascade requires the involvement of AC1C4. BC3, BC4, BCC5, C9, DC2, C4. Answer. CC5, C9. 3. CSIR-NET June 2018. Which of the following complement proteins is primarily responsible for the formation of the C3 convertase in the classical pathway? AC1. BC4, BC2, ACC3, BDC5B. Answer. BC4, BC2A. Conclusion. The complement system is an essential component of the innate immune system, providing a rapid and effective response to infection. Through its cascade of activation, the complement system promotes opsonization, inflammation, and cell lysis, contributing to the defense against pathogens. Regulation of the system is critical to avoid host tissue damage and prevent autoimmune conditions. Understanding the complement system is essential for appreciating its role in both normal immune responses and disease mechanisms, making it a key area for CSIR-NET life sciences preparation. Let me know if you'd like to move to the next topic or need more details on any section. You said. Primary and secondary immune modulation, without table ChatGPT said. Here is your CSIR-NET JRF Life Sciences level lecture on primary and secondary immune modulation, written in maximum detail, without tables, and tailored to the CSIR-NET exam. This lecture also includes 2 to 3 previous year questions with answers to aid your exam preparation. Primary and secondary immune modulation, CSIR-NET Life Sciences lecture introduction. Immune modulation refers to the regulation or adjustment of immune system activity. It can occur at various levels, from altering the activity of individual immune cells to broader adjustments in the entire immune system's response. Immune modulation is vital for maintaining immune homeostasis and ensuring appropriate immune responses. It is crucial in the context of infections, autoimmunity, cancer, and other immune-related conditions. Immune modulation can be classified into primary immune modulation and secondary immune modulation, which differ based on the mechanisms involved in the context in which they occur. Primary immune modulation. Primary immune modulation refers to the regulation of immune responses during the initial encounter with pathogens or foreign antigens. This modulation typically involves innate immune cells, such as macrophages, dendritic cells, and natural killer NK cells, which play key roles in recognizing and responding to pathogen-associated molecular patterns (PAMPs) and damage-associated molecular patterns (DAMPs). Activation of pattern recognition receptors (PRRs). The first step in primary immune modulation is the detection of foreign entities, pathogens, allergens, or damaged cells through PRRs, such as toll-like receptors (TLRs) and nod-like receptors (NLRs). These receptors are found on the surface of innate immune cells and can recognize specific molecular structures unique to pathogens or stressed cells. Cytokine release. Following PRR activation, immune cells secrete various pro-inflammatory cytokines like interleukins, IL-2, tumor necrosis factor alpha, TNF-alpha, and interferons, IFNs. These cytokines serve as signals to recruit additional immune cells, such as neutrophils, monocytes, and T cells, to the site of infection or inflammation. Complement activation. The complement system is another crucial component of primary immune modulation. It is activated by the presence of pathogens and contributes to the elimination of pathogens through opsonization, cell lysis, and inflammation. Inflammatory response. The innate immune cells also contribute to local inflammation by promoting the release of inflammatory mediators, which increase vascular permeability, allowing immune cells and antibodies to reach the infection site. This process is essential for the early control of infections. Examples of primary immune modulation. In response to a viral infection, dendritic cells will capture viral particles, process them, and release pro-inflammatory cytokines such as type 1 interferons, which initiate an antiviral immune response and promote the activation of adaptive immune cells. Upon injury, macrophages release TNF-alpha, which triggers local inflammation and immune cell recruitment. Secondary immune modulation. Secondary immune modulation refers to the regulation of immune responses after the primary immune activation, often through interactions between adaptive immune cells, such as T cells and B cells, and antigen-presenting cells, APCs, such as dendritic cells. Secondary immune modulation generally occurs in lymphoid organs, such as lymph nodes or the spleen, and is associated with the activation of memory cells and the fine-tuning of immune responses. Adaptive immune activation. During secondary immune modulation, T helper cells, TH cells, play a central role in coordinating the immune response. These cells can influence the function of cytotoxic T lymphocytes, CTLs, B cells, and other immune cells through cytokine secretion. For example, TH1 cells activate macrophages and CTLs, while TH2 cells promote B cell activation and antibody production. Regulatory T cells, TREGs, in secondary immune modulation. TREGs help maintain immune tolerance and prevent excessive inflammation. They can suppress the activation of effector T cells, thus ensuring that immune responses are regulated and do not result in autoimmune reactions. Memory formation. One of the key features of secondary immune modulation is the establishment of immune memory. After an initial immune response, memory B cells and T cells persist in the body. These cells allow for a quicker and more robust response upon subsequent exposure to the same pathogen. This phenomenon is the basis of vaccination. Cytokine networks. In secondary immune modulation, cytokines such as interleukins IL-4, IL-6, IL-10, IL-12 play essential roles in shaping adaptive immune responses. For example, IL-4 promotes Th2 responses and IgE production, which is important in allergic responses. IL-10 is an immunosuppressive cytokine that helps maintain immune balance by downregulating excessive immune responses. Examples of secondary immune modulation. After vaccination with an inactivated pathogen, memory B cells are formed and can rapidly produce antibodies upon re-exposure to the pathogen. Tregs control the potential for immune overactivation following an infection to prevent damage to host tissues. Key differences between primary and secondary immune modulation, timing and context. Primary immune modulation occurs during the initial immune response to a pathogen, while secondary immune modulation occurs upon subsequent exposures or after the resolution of a primary immune response. Involved cells. Primary immune modulation is primarily mediated by innate immune cells, e.g., macrophages, dendritic cells, NK cells, whereas secondary immune modulation involves the activation and regulation of adaptive immune cells, e.g., T cells, B cells, Tregs. Outcomes. Primary immune modulation results in the activation of the initial defense and inflammation, while secondary immune modulation leads to the enhancement of immune memory and the fine-tuning of responses to prevent overactivation. Cytokine production. In primary modulation, the main cytokines include TNF-alpha, IL-1, and interferons, which mediate inflammation and early defense. In secondary modulation, IL-10, IL-4, and IL-12 are more commonly involved, regulating immune responses and facilitating adaptive immunity. Clinical relevance of immune modulation. Primary immune modulation. It is essential for the initial defense against infections, particularly in the early stages of immune recognition and response. Defects in primary immune modulation, such as mutations in pattern recognition receptors (PRRs), can lead to increased susceptibility to infections and impaired immune responses. Secondary immune modulation. Essential for long-term immunity and the establishment of immune memory, making it critical for the efficacy of vaccines. Overactive secondary immune modulation can contribute to autoimmune diseases, where the immune system erroneously targets host tissues, e.g., in diseases like rheumatoid arthritis or multiple sclerosis. Immunosuppressive therapies, e.g., the use of corticosteroids or biologics targeting cytokines, are used to modulate secondary immune responses, especially in conditions such as autoimmune disorders or after organ transplants. CSIR Net previous year questions. 1. CSIR. Which of the following is a characteristic feature of secondary immune modulation? Initiation of inflammation. B. Formation of immune memory. C. Activation of pattern recognition receptors. D. Phagocytosis of pathogens. Answer. B. Formation of immune memory. 2. CSIR Net June 2019. What role do regulatory T cells, Tregs, play in secondary immune modulation? A. Promoting inflammation. B. Enhancing pathogen clearance. C. Suppressing immune responses. D. Activating memory B cells. Answer. C. Suppressing immune responses. 3. CSIR Net June 2018. Which cytokine is primarily involved in promoting Th1 responses during secondary immune modulation? A. IL-4. B. IL-6. C. IL-10. D. IL-12. Answer. D. IL-12. Conclusion. Both primary and secondary immune modulation are essential for ensuring a balanced and efficient immune response to infections and other challenges. Primary immune modulation provides an immediate defense through innate immune mechanisms, while secondary immune modulation fine-tunes the adaptive immune response, leading to the formation of immune memory and long-lasting protection. Understanding these processes is vital for interpreting immune responses in health and disease, and for developing therapies for infectious, autoimmune, and inflammatory diseases. Let me know if you need further clarification or want to move to the next topic. You said. Toll-like receptors. ChatGPT said. Toll-like receptors TLRs. CSIR Net JRF Life Sciences Lecture Introduction. Toll-like receptors TLRs are a family of pattern recognition receptors PRRs that play a critical role in the innate immune system. These receptors are involved in the recognition of pathogen-associated molecular patterns (PAMPs) and damage-associated molecular patterns (DAMPs), which are present on pathogens and damaged host cells, respectively. TLRs are essential for initiating the immune response to infections and for maintaining tissue homeostasis. Structure and location of toll-like receptors. TLRs are transmembrane proteins characterized by an extracellular leucine-rich repeat LRR domain, which is responsible for recognizing PAMPs, and an intracellular toll interleukin-1 receptor TIR domain, which is involved in signal transduction. TLRs are located on the surface of various immune cells, such as macrophages, dendritic cells, and B cells, as well as in the endosomal membranes of innate immune cells. Key TLRs and their locations. TLR1, TLR2, TLR4, TLR5, TLR6, these are typically found on the cell surface. TLR3, TLR7, TLR8, TLR9, these are located on the endosomal membrane within the cytoplasm. Mechanism of action. The primary function of TLRs is to recognize specific molecular patterns found on pathogens, bacteria, viruses, fungi, etc., or damaged host cells and trigger immune signaling pathways. The recognition of these patterns by TLRs results in the activation of downstream signaling cascades that lead to the production of pro-inflammatory cytokines, the activation of immune cells, and the induction of an adaptive immune response. Signaling pathway. Ligand binding. When a pathogen or damaged cell component, like lipopolysaccharides (LPS) from bacteria, viral RNA, or unmethylated CpG DNA, binds to a TLR, the extracellular LRR domain recognizes these patterns. Activation of TIR domain. Binding of the ligand to the LRR domain induces a conformational change that activates the TIR domain. Recruitment of adapter proteins. The TIR domain recruits various adapter proteins, including MYD88, myeloid differentiation factor 88, TRIF TIR domain containing adapter-inducing interferon beta, and TICAM1 TIR domain containing adapter molecule 1. These adapters further activate intracellular signaling pathways. Activation of NF-kappa B pathway. The MYD88 dependent pathway is the most common and leads to the activation of NF-kappa B, nuclear factor kappa B, a transcription factor that triggers the production of inflammatory cytokines, such as TNF-alpha, IL-1, and IL-6. This response helps initiate acute inflammation. Type 1 interferon production. The TRIF-dependent pathway leads to the activation of IRF3, interferon regulatory factor 3, promoting the production of type 1 interferons, IFN-alpha and IFN-beta. These interferons are crucial for antiviral immunity. Activation of adaptive immunity. The release of cytokines and interferons further recruits and activates adaptive immune cells like T cells and B cells, enhancing the immune response. Types of TLRs and their specific ligands. Each TLR is specialized in recognizing a distinct class of PAMPs or DAMPs. Here are some major TLRs and the ligands they recognize. TLR1 and TLR2 recognize bacterial lipoproteins and lipoteichoic acids. They work in tandem to detect gram-positive bacteria. TLR3 recognizes double-stranded RNA, dsRNA, a pattern typically found in viruses, especially during viral infections. TLR4 primarily recognizes lipopolysaccharide, LPS, from the outer membrane of gram-negative bacteria. TLR4 is essential for the recognition of bacterial infections. TLR5 recognizes flagellin, a component of the bacterial flagellum. TLR6 pairs with TLR2 to recognize diacylated lipoproteins from gram-positive bacteria and fungi. TLR7 recognizes single-stranded RNA, ssRNA, which is common in many viruses, particularly RNA viruses. TLR8, similar to TLR7, TLR8 recognizes ssRNA from viruses. TLR9 recognizes unmethylated CpG DNA, which is common in bacteria and viruses. TLR10, its function is less understood, but it is believed to play a role in recognizing pathogen-derived molecules and regulating immune responses. Functions of TLRs in immune responses. Innate immunity. TLRs are a crucial part of the body's first line of defense against infections. By recognizing and binding PAMPs, they enable the rapid activation of immune responses that can contain infections before the adaptive immune system is fully activated. Induction of inflammation. TLR activation results in the secretion of pro-inflammatory cytokines such as TNF-alpha, IL-6, and IL-1, which mediate the inflammatory response to clear pathogens. However, excessive TLR signaling can also lead to chronic inflammation, contributing to diseases like autoimmune disorders or sepsis. Regulation of adaptive immunity. Through the activation of dendritic cells and macrophages, TLRs play a key role in antigen presentation to T cells. This results in the induction of T helper Th cell differentiation and antibody production by B cells, thus bridging the innate and adaptive immune systems. Host defense. TLRs help recognize and initiate immune responses against a wide variety of pathogens, including bacteria, viruses, fungi, and parasites. This makes them critical in controlling infections in the early stages. Clinical relevance of toll-like receptors. Infections. TLRs are central to the recognition and defense against a variety of pathogens. Mutations or deficiencies in TLR genes can lead to increased susceptibility to infections, including bacterial, viral, and fungal infections. Autoimmunity. Aberrant TLR signaling can lead to autoimmune diseases. For instance, TLR7 and TLR9 have been implicated in diseases like systemic lupus erythematosus, SLE, where the immune system mistakenly attacks the body's own tissues due to a heightened response to self-DNA. Cancer. TLRs can also play a dual role in cancer. On one hand, they help mount an immune response against tumors. On the other hand, chronic TLR activation may contribute to tumor progression and immune evasion, making TLRs potential targets for cancer therapies. Therapeutic targeting. TLRs have become therapeutic targets in various clinical settings. TLR agonists like imiquimod for topical treatment of basal cell carcinoma and TLR antagonists to block excessive inflammation in autoimmune diseases are under investigation as potential treatments. CSIR-NET previous year questions. 1. CSIR-NET June 2019. Which of the following TLRs recognizes lipopolysaccharide, LPS? ATLR1. BTLR4 circuit TLR5 DTLR9 answer, BTLR4. 2. CSIR-NET DEC 2018. TLR3 is primarily involved in the recognition of which of the following pathogen-associated molecular patterns PAMPs? A. Flagellin. B. Double-stranded RNA, dsRNA, C. Lipopolysaccharide, DCPG-DNA. Answer, B. Double-stranded RNA, dsRNA. 3. CSIR-NET June 2020. Which TLR is associated with the recognition of viral RNA in the endosome? ATLR2. BTLR4 circuit TLR7 DTLR9 answer, CTLR7. Conclusion. Toll-like receptors TLRs are pivotal in the immune system's ability to recognize and respond to a wide variety of pathogens. Their role in triggering innate immune responses and bridging the innate and adaptive immune systems makes them essential for effective immune defense. Understanding the structure, function, and signaling pathways of TLRs is crucial for developing targeted therapies for infections, autoimmunity, and cancer. Let me know if you need more details or want to move on to another topic. You said. Cell-mediated effector functions, ChatGPT said. Cell-mediated effector functions, CSIR-NET JRF Life Sciences Lecture Introduction. Cell-mediated immunity, CMI, is a crucial arm of the adaptive immune system that involves T lymphocytes, T cells, and is primarily responsible for defending the body against intracellular pathogens, such as viruses, bacteria, fungi, and tumor cells. Unlike humoral immunity, which is mediated by antibodies, cell-mediated immunity involves direct actions of immune cells like cytotoxic T cells, CTLs, helper T cells, Th cells, macrophages, and natural killer NK cells. The effector functions of cell-mediated immunity refer to the various mechanisms by which T cells and other immune cells exert their roles in eliminating infected or abnormal cells, modulating immune responses, and maintaining immune surveillance. Key players in cell-mediated effector functions: cytotoxic T lymphocytes, CTLs. CD8+ T cells are the main effectors in cytotoxic responses. They directly recognize and kill infected cells or tumor cells expressing foreign or altered antigens presented by MHC class I molecules. Helper T lymphocytes, Th cells. CD4+ T cells provide assistance to other immune cells, such as macrophages, B cells, and CTLs, through the secretion of cytokines. They are categorized into different subsets based on the cytokines they produce, e.g., Th1, Th2, Th17. Macrophages. These are professional phagocytes that engulf and destroy pathogens and present antigens. Upon activation by Th1 cells or through TLR signaling, macrophages release pro-inflammatory cytokines and further enhance immune responses. Natural killer NK cells. NK cells are innate immune cells that recognize and kill cells that are virus-infected or tumorigenic through a mechanism that is independent of antigen presentation. Mechanisms of cell-mediated effector functions. 1. Cytotoxic T lymphocyte, CTL, mediated killing. Cytotoxic T cells recognize specific antigens presented by infected cells via MHC class I molecules. Once the antigen is recognized, CTLs are activated to exert their effector function through two primary mechanisms. Perforin granzyme pathway. Upon activation, CTLs release perforin, a pore-forming protein, which inserts into the membrane of the target cell. Granzymes, serine proteases, enter the target cell through these pores and induce apoptosis by cleaving proteins involved in cell survival. Phos-Facil pathway. CTLs express Phos ligand Facil on their surface, which binds to Phos CD95 receptors on target cells, leading to a signaling cascade that induces apoptosis. These mechanisms effectively eliminate infected or tumor cells by inducing programmed cell death apoptosis. 2. Helper T cell mediated functions. Helper T cells play a central role in coordinating immune responses by secreting cytokines that influence the activities of other immune cells. Th1 cells. Th1 cells primarily produce interferon gamma, IFN gamma, which activates macrophages and promotes the development of cytotoxic T cells. Th1 cytokines enhance cellular immunity and are important in the defense against intracellular pathogens, e.g., viruses and mycobacteria. Th2 cells. Th2 cells secrete IL-4, IL-5, and IL-13, which stimulate B cells to produce antibodies, especially IgE, and promote the defense against extracellular parasites, e.g., helminths. Th17 cells. Th17 cells produce IL-17, which is important for the recruitment of neutrophils and other inflammatory cells to sites of infection, particularly in fungal infections and autoimmune diseases. Regulatory T cells, TREGs. TREGs are critical for maintaining immune tolerance and preventing autoimmunity. They suppress the activation of autoreactive T cells and modulate the immune response to prevent excessive inflammation. 3. Macrophage activation. Macrophages play a pivotal role in cell-mediated immunity through their ability to recognize pathogens and present antigens to T cells. Activated macrophages exhibit enhanced killing capacity against pathogens and are induced to produce pro-inflammatory cytokines such as TNF-alpha, IL-1, and IL-6. This inflammatory environment helps recruit other immune cells, including neutrophils and lymphocytes. Classically activated macrophages M1 are induced by IFN gamma, often from Th1 cells, and are involved in microbial killing, antigen presentation, and the production of inflammatory cytokines. Alternatively activated macrophages M2 induced by IL-4 and IL-13 from Th2 cells are involved in tissue repair, anti-inflammatory responses, and resolution of inflammation. 4. Natural killer NK cells. NK cells are critical in the innate immune response to viruses and tumors. They recognize and kill cells that do not express MHC class I molecules, which is a hallmark of infected or malignant cells. NK cells express activating receptors such as NKG2D that bind to stress-induced ligands on infected or transformed cells, triggering the release of cytotoxic granules containing perforin and granzymes. Cytokines and their role in cell-mediated effector functions. Cytokines are signaling molecules that are produced during immune responses. They influence the activation, differentiation, and function of immune cells. IFN gamma, produced mainly by Th1 cells and CTLs, it activates macrophages, enhances antigen presentation, and induces the Th1 differentiation pathway. TNF-alpha, produced by macrophages, T cells, and NK cells, it plays a key role in inflammation and the killing of tumor cells and infected cells. IL-2, secreted by CD4 plus T cells, IL-2 promotes the proliferation and survival of CD8 plus T cells and is critical for T cell expansion during immune responses. IL-12, produced by macrophages and dendritic cells, it induces Th1 differentiation and promotes IFN gamma production. IL-4 and IL-5, produced by Th2 cells, they stimulate B cell antibody production and eosinophil activation during responses against helminths and allergic reactions. Clinical relevance of cell-mediated effector functions. Immunodeficiencies. Defects in the T-cell-mediated immune response can lead to severe combined immunodeficiency, SCID, where patients have impaired defense against intracellular pathogens. Autoimmune diseases. Dysregulated cell-mediated immunity can result in autoimmune diseases. For instance, type 1 diabetes is caused by CTL-mediated destruction of pancreatic beta cells. Cancer. Tumors can evade immune surveillance by downregulating MHC class I expression or secreting immunosuppressive cytokines. Enhancing CTL responses or utilizing checkpoint inhibitors, like PD-1, PD-L1 inhibitors, is an emerging therapy in cancer immunotherapy. Infections. Cell-mediated immunity is vital for controlling viral infections, e.g., HIV, influenza, and intracellular bacterial infections, e.g., Mycobacterium tuberculosis. CSIR-NET previous year questions. 1. CSIR-NET June 2019. Which of the following cytokines is primarily responsible for activating macrophages during a Th1 response? AIL-4. BL10 circa IFN gamma DL17 answer. CIFN gamma. 2. CSIR-NET DEC 2020. Cytotoxic T lymphocytes, CTLs, kill target cells through which mechanism? A perforant granzymes. B phagocytosis C activation of NK cells D antibody-dependent cytotoxicity answer. A perforant granzymes. Conclusion. Cell-mediated immunity is crucial for the defense against intracellular pathogens, tumor cells, and the regulation of the overall response. The effector functions of CTLs, helper T cells, macrophages, and NK cells involve complex interactions that lead to pathogen elimination, tumor destruction, and the maintenance of immune homeostasis. Understanding these functions is essential for developing therapeutic strategies for infections, cancer, and autoimmune diseases. Let me know if you'd like to explore more details or another topic. You said,